we would like to introduce a visualization framework specifically designed to facilitate RNA expression analysis, motivated by the questions usually encountered in glycobiology, that is, how the expression of a set of genes are different from one tissue to another, and how their co-expression differs in those tissues. We start by choosing a dataset. In this current version, the application comes with 3 versions of the pre-processed RNA-seq data from the GTX project. These datasets are the result of alternative processing workflows on the same set of experiments. The first step, expression pattern, allows us to juxtapose the expression of several genes simultaneously and overlay the profiles of multiple tissues. We start by selecting some genes of interest, either by typing the gene names or more conveniently selecting a gene set. Then limit the samples to some organs of interest or more specific tissue types. The application will then try to retrieve the data of interest, plot the expression levels on a parallel coordinate plot, display the detailed information about the samples in the table, and summarize the count of samples on several interesting phenotypes. Each line in the plot here corresponds to one sample, and the position on its y-axis represents the transcript abundance of the corresponding gene. Interacting with the plot will result in synchronized response in the other components. The data table will highlight the selected samples, and the bar charts will summarize the number of samples in each category by count or by percentage. We can also select samples from the table using some criteria of interest to achieve the same effect as brushing on the parallel. The second tab, correlation, allows us to visualize the pairwise correlation of gene expression level within a group of samples. We can select genes and samples in a similar fashion as previously, using gene names, selecting gene sets, and selecting tissue types. We can also quickly transfer the genes from the expression tab over here by clicking at this button. The Pearson correlation between every pair of genes will be calculated and displayed as a matrix in which the square is colored corresponding to the level of correlation. The color goes from dark red to white to dark blue, corresponding to the correlation coefficient from -1 to 0 to 1. So we know that Pearson correlation cannot capture nonlinear relations. So to investigate the true relation of each pair, we can click on the matrix to reveal a scatter plot of one gene against another. And in general, scatter plots are the most telling graphs to uncover pairwise relation. Many times, an interesting structure can be hidden by the correlation coefficient, and in such cases, we can see if the structures align with any known partitioning of data, such as organ or gender, by highlighting them with different colors.